In this video, we'll talk about how LIMS Nucleus handles replication. You can read about replication on the replication page. Samples and targets are each replicated independently. Let's define two terms, sample and target. Sample is what we are tracking through multi-well plates. It might be antibodies, small molecules, siRNAs, maybe plasmids, maybe bacterial stocks. Targets are the entities to which the samples are applied. So for example, in an ELISA, an antibody is applied to a protein that is bound to the plate. Maybe a small molecule is being applied to an enzyme in an enzyme inhibition assay, and possibly siRNAs are being applied to cells in a knockdown experiment. You could also apply plasmids to cells in an expression assay. Another term that needs clarification is quadrants. In LIMS Nucleus, a quadrant in a 384 well plate are the wells that are accessed by one transfer of a fixed tip 96 channel pipetter. So if the upper left tip goes into A1, every other well across columns and every other well down rows is quadrant 1. You can see that more clearly here where quadrant 1 wells are in black. Quadrant 2 is 1 over. Quadrant 3 has B1 as the first well, and quadrant 4 has B2. So in LIMS Nucleus, we are always working in a Z pattern. If you are transferring 4 96 well plates into a 384 well plate, plate 1 goes into quadrant 1, 2 into quadrant 2, and so on. Plate 4 into quadrant 4 in a Z pattern. When reformatting 96 well plates into a 384 well plate, you have multiple options. Here I'm showing 4 different plates being combined into a single 384 well plate. Plate 1 goes into quadrant 1, plate 2 into quadrant 2, and so on in a Z pattern. That transfer is represented diagrammatically right here. Each plate, each unique 96 well plate is represented by a different color. 4 96 well plates are combined into one 384 well plate. Since each 96 well plate is represented once, the samples in that plate have a replication of 1, hence 1s for sample. Another possibility is that you have 2 plates that you are transferring in duplicate to the 384 well plate. The 96 well plates are represented by the colors. Plate 1 is black, plate 2 is white. Each are transferred twice. Plate 1 into quadrant 1 and 2, plate 2 into quadrant 3 and 4. And so now each sample is represented twice, hence 2s. Sample replication in LIMS Nucleus is always horizontal within a row. A third option is that you have a single plate, single 96 well plate that is transferred into all 4 quadrants of the 384 well plate. Now each sample will be represented with an N of 4. Note that when using LIMS Nucleus, you must use one of these 3 patterns of sample replication, either N of 1 with different plates in all 4 quadrants, N of 2 with replicates horizontal across columns, or a single plate in quadruplicate. Other patterns like 3 and 1 are not supported. Target replication follows similar patterns. Targets will be the possibly antigen that is coated on the bottom of the plate or cells that are deposited in the well. Targets are represented either once, an N of 1, or they're represented in duplicate. The difference now is that duplicate targets are in the same column. They are vertical. Or the target is represented, the same target is represented in all 4 quadrants. So given the 3 patterns that are supported for sample replication and the 3 patterns supported for target replication, when combined, there are 5 different combinations that are supported by LIMS Nucleus. You must use one of these combinations for LIMS Nucleus to work for you. These 5 different combinations are the layouts that are automatically generated for you when you create a layout in LIMS Nucleus. See the layouts video for more information. This first combination, 1S4T, is when you have a large number of plates that you want to screen on a single target. Assay replication will be an N of 1. If you have 2 targets to screen, you would use 2S2T, maybe a positive and a negative control. Here, though the 96 well sample plates are represented twice, one copy is screened on target 1 and the other on target 2 for a final assay replication of 1. To screen a single target with samples in duplicate, you would use 2S4T. 4S1T and 4S2T are to screen many targets, either with an N of 1 or in duplicate. In this slide, we'll show how the different combinations of layouts can be used in a screening campaign. The blue arrow represents a reformatting operation, that is, combining multiple 96 well plates into 384 well plates. The red arrow is a rearray or cherry picking operation. This will be a 3-step campaign, with the 3rd step having 2 options. Realize that this schematic represents the 4 destination wells in a 384 well plate for any well, source well, in the 96 well plate. For example, here are those 4 wells, quadrant 1, 2, 3, 4, that represent the destination of the sample from the source plate, well E12. The first step of our screen is the high throughput screen. We have a large number of plates, 1,096 well plates, that will be reformatted into 250 384 well plates. 4 plates, 496 well plates per 384 well plate. We will be screening antibodies on the antigen human CD71, the transferrin receptor, looking for transferrin receptor specific antibodies. We run our assay and then we cherry pick the hits from the 1,000 plates and find that we have a total of 8 96 well plates worth of hits. Those 8 plates will fit into 2 384 well plates for follow-up screening, but we'd like to screen in duplicate. So each one of the 96 well plates gets transferred in duplicate for a total of 4 384 well plates. We run the assay on the 4 plates, cherry pick the hits, and find that we have a single plate of hits, single 96 well plate of hits. Now we want to screen those hits on human, murine, and cyno CD71, as well as a negative control protein. We are interested in a human therapeutic, but we also have mouse and monkey models in which we would like to test our therapeutic. So we will favor hits that are cross-reactive with murine and cyno CD71, but don't bind the negative control. If we run our single 96 well plate in quadruplicate on a 384 well plate coated with 4 different antigens, we will get an N of 1. But we could generate replicates by running multiple plates. So if we ran 3 plates in this layout, we'd have triplicates with our triplicates across plates. Another option would be to coat only 2 of the antigens in duplicate. This would give us within plate replicates. We'd have to run this experiment twice, with the second run being the negative control and cyno CD71. And likewise, we could run each of these plates multiple times, generating higher order replication. Now, with an understanding of the replication notation, we can look at the set of layouts associated with the source layout, layout 1. Layout 1 involves 4 controls in column 12. If we rearray that plate so that sample has an N of 1 and target an N of 4, that's layout 2. So the pattern of controls does not change. But now we can see that for an N of 1, each of the 4 plates is transferred to each quadrant of the 384 well plate, and this pattern is across the plate. For target replication, only one target is used, target 1, that is in all 4 quadrants of the plate. If we look at the layout where both sample and target are an N of 2, again, controls don't change, but the sample replication, we have 2 plates, plate 1 and plate 2, each replicated twice horizontally across the rows. For target, we're using 2 targets, target 1 and target 2, replicated vertically down columns. Layouts and replication work together to define the contents of your plates. Be sure you understand how LIMS Nucleus handles replication and layouts before you set up your assay. If you have questions as to the suitability of LIMS Nucleus for your process, contact us at info@labsolutions.com.